Hi, Assalamualaikum and hi everyone. So today we are going to discuss about biochemical mechanism of neem, or known as Azadirachta indica, as an insect repellent. This is our group members, which is me myself, Nur Anisa binti Isha, and my colleague Sharifa Irin Batrisha. So what is neem? Neem, also known as Azadirachta indica, which is a native to Indian and Southeast Asia. It also known for strong antimicrobial insecticidal and ecological benefits. Why is it important? Neem is important because it can produce unique bioactive secondary metabolites that influence ecological interactions.
There are three concept overview for this presentation. First off is where the neem compounds are located in the plant, and another is the core biochemical pathway it's producing, and the specific mechanism by which the azadirachtin affects the insect physiology.
So there are three main biochemical compounds, which is azadirachtin, limbin, and also selenin. So now let's move on to the three main biochemical actions.
One of the main biochemical action, which is the anti-feeding mechanism, where the azadirachtin, a limonoid synthesized via mevalonate pathway. This bioactive compound blocks the gustatory receptor, which could deter the feeding of the insect, and is also the midgut of the epithelial cells also disrupted, and reducing the digestive enzyme activity that could cause the starvation. And ecdysone hormones, which could help in insect molting, also affected, and it also affected its normal development. And fun fact, actually there are this mechanism acts over the 200 insect species and qualify neem as a powerful natural plant defiance compound. And next is the hormonal disruption, which is the insect growth regulation. The azadirachtin also interferes in insect molting hormone system, which it blocks the PTTH release from the insect brain that could prevent the activation of the prothoracic gland, and the synthesis of ecdysone is also reduced, causing the insect cannot produce enough molting hormone, and it also blocks the ecdysone receptor, and thus stopping the action of the 20-hydroxyecdysone. And any protein and chitin synthesis for the new cuticle are also disrupted, causing the molting failure. So the outcome is the insect cannot molt and remain stunted and die. Lastly is the repellent action, where the plant volatiles such as azadirachtin, citronellal, geraniol, and limonene could act as insect repellent, where this molecule could evaporate easily and form a volatile chemical barrier around the plant. So when the insect interfere into the chemical barrier, the insect's olfactory receptors are interfered, which prevent it from detecting any host cues. And this compound also block and confuse odor binding proteins and disrupt the signal transmission, and thus, the insect will fail to recognize the plant as a food or any suitable site. This, in result, will reduce landing, feeding, and stopping any egg laying on the treated plant or any surface.
Now let's look to the case study, which is the larvicidal activity of neem oil Azadirachta indica formulation against the mosquitoes. So there are studies stated that neem oil formulation they use caused a very high reduction in larval density in all tested breeding sites. According to this result, in the entire breeding sites, larval density reduction reached 100% by day 7 of the treatment.
So one of the uses of neem in Malaysia is the snail control, where the neem extract and the neem oil are used to control the golden apple snail, a major rice pest in Malaysia. So the azadirachtin and limonoids act as a molluscicides that reducing nail feeding and the reproduction.
Next, neem also used as a soil-based biocontrol.
Malaysia is the mosquito larvicide control, where the neem seed oil and azadirachtin show a strong larvicide activity against the Aedes aegypti and the Aedes albopictus. And lastly is the biocontrol in Malaysian agriculture, where it is widely used as the biopesticide against a major crop pest. In conclusion, the neem itself has been protected through multiple biochemical defenses, which is the anti-feeding, repellent and hormone disruption. So the azadirachtin mainly, the key lemnoids interfere in insect feeding behavior, growth hormone and also the sensory detection. So more biochemical pathways show how the plant use their secondary metabolite to survive in herbivory in ecosystem. And the neem's natural defenses compound could highlight a connection between the molecular mechanism and the ecological function.
So this is our references, and that's all from us. Thank you for watching.